Win India webinar on an interesting topic, that is novel transplant biomarkers. As we all know, diagnostics play a significant role in the management of transplant patients, in the diagnosis as well as in management. So we have a long way to go regarding the diagnostics. So we have here today an interesting webinar on this topic. To moderate this topic, we have with us Dr. Priya Pais, who is a pediatric nephrologist from St. John's National Health Academy of Sciences. Along with her joining is Dr. Arpana Iyengar, who is Professor, Pediatric Nephrology, St. John's Institute of Medical Sciences. So without much ado, I welcome the speaker as well as the moderators to continue the session. Over to you, Dr. Priya.
Thank you, Dr. Manjusha. It is my great pleasure to welcome and introduce Dr. Vikas Dharnidata to you all. He is the Chief of the Division of Pediatric Nephrology, Hypertension and Pheresis at Washington University in St. Louis. He is also the Vice Chair for Clinical Investigation in the Department of Pediatrics and a tenured Professor of Pediatrics. He holds the Alexis F. Hartman Senior MD Endowed Chair in Pediatrics, and he's obtained his MBBS and MD from Topiwala National Medical College and Bombay University, and an MPH in Biostatistics from the University of Florida. He completed his pediatric nephrology fellowship training at Children's Hospital and Harvard University in Boston. Dr. Vikas is a keen researcher, and as a clinical and translational researcher, he has created and led multidisciplinary teams that perform many different kinds of research, ranging from translational metagenomic sequencing projects to large database integrations to compare effectiveness in clinical biomarker studies. His particular areas of focus are, of course, clinical biomarkers in transplant, post-transplant infections and malignancies, in particular PTLD and BK virus nephropathies, very important topics in pediatric kidney transplantation. He has published more than 220 papers, including in the New England Journal of Medicine, the Lancet, and Cancer Medicine. So we welcome Dr. Vikas, and we're really looking forward to hearing you speak today on novel transplant biomarkers.
Thank you, Dr. Priya, for that very, very kind introduction. And thank you to the Women in Nephrology India for inviting me to present this topic to you. And I will now pull up my slides. I don't know if you can see any slides. I'm seeing a black screen here, so give me 1 second. We'll try it again.
That was the last slide of your presentation when we had done the tech check. I think you can go to the first slide. Yes.
Yeah, it should be on the first slide is what I saw. And so let's see if I do that. So maybe I had to do it again.
No, now we are able to see. Yeah.
Yes, now we see it.
Okay, thank you. So these are my disclosures. I do get grant support from different branches of National Institutes of Health, and then also from CareDx, which is relevant to some of the topics I will talk about. And I have been a consultant to Atara, Sanofi, Transplant Genomics, MedinCell, of which Transplant Genomics is also relevant to what I will talk about today. So on the left of this square box, what you see is what we all hope to have, a normal or pristine allograft, which functions well and looks normal on histology. On the right, what you see is how we have managed so far to look for clinical alterations. The most common thing that we see is an elevation in serum creatinine or GFR loss. And serum creatinine elevations can occur due to a variety of reasons, hypovolemia or tacrolimus toxicity, urinary tract infection. The big one that we want to always catch early and treat and reverse early is an acute transplant rejection, immunologically mediated. So the problem is that creatinine rises late in the course of acute rejection. And by the time you see an elevation in creatinine, the rejection has already been going on for a while subclinically in the tissue level. So the chance that you can fully reverse it becomes less. There's going to be likely some residual damage, even if you do treat an acute rejection. And then that sets up a sequence of events which leads to chronic allograft dysfunction and earlier graft loss. So what can we do to change that time progression? And if you go to the middle box, what we have been doing so far is trying to pick up these morphological alterations at an earlier stage, typically by what we call protocol or surveillance biopsies, which are done at fixed time points, even if the serum creatinine is normal. And if we see subclinical rejection, we can try and treat that. Harder to treat subclinical interstitial fibrosis or tubular atrophy. What the novel transplant biomarkers are trying to do in part is to take that window even further to the left in the light purple, where we're looking at molecular markers, which could be gene transcriptomics, but could also be other protein markers or single molecules that we think will elevate prior to an acute rejection and will offer a greater and earlier sensitivity while also being specific. So this table is from a review paper that Dr. Andrew Malone and myself had published a few years ago, and it shows you the various types of what we call as less invasive biomarkers. The term less invasive is preferred to non-invasive when, particularly when you have blood tests, it still involves a needle stick for that. Whereas if you have a urine test, then the biomarker would be truly a non-invasive marker. So the various types include different antibody assays, which we're using right now, donor-specific assays, but could also include T-cell reactivity assays, gene expression panels, which we'll talk about, chemokines, then microRNAs, donor-derived cell-free DNA, which also I will discuss in detail, and then some other tests. Some of you may be familiar with the older immunoassay, which was a measure of ATP levels from CD4 positive lymphocytes. And then there are a variety of other single molecule markers such as IDO or fractalkine, soluble CD30, 103. Most of these are in peripheral blood. A few of these are in urine. What you see here in this column are the area under the curves for each of these tests. And generally for area under the curve, you'd like it to be closer to 1.0. The closer you can get, the better, more accurate that test is, both in terms of sensitivity and specificity. And you can see that many of these tests run in the 0.7 to 0.8 range. Some of them have a range that could get into the 0.9 and above, but these are the median AUCs for many of these tests. And they all have some peculiar characteristics, which I won't go into too much detail now, but these tests, some work better than others for a particular type of rejection or for infection, or they represent only one compartment of the immune system, such as just the humoral compartment or just the cellular compartment. Among the gene expression panels, these are ones that are commercially marketed right now in the US. So there's this AlloMap, which is actually a heart transplant panel. Now there's an AlloMap kidney. Then there is a K-sort panel, then a Quest renal transplant monitoring panel, which now goes under a different name called EuroMap, and that is actually part of CareDx panel 2. And then a different panel, GOCAR, which for rejection the way the first three panels are, but looking to predict fibrosis. Now, all of these are, you don't have to memorize these are all the different genes listed over here. What was interesting to us when we were looking at these different panels, we went actually to the gene nomenclature committee to see what some of these genes, they have different names. So maybe some of them were the same gene, but just under a different name. But they truly, each of the genes here versus each of those here, here, and here are all unique in the respective panel. There is not actually any overlap between any gene in any of these panels, even though we think of the immunological pathways across rejection to be common, whether it's kidney, heart, liver, or lung transplant. So just very briefly about how that immunology works. When you place an allograft, the allograft is going to have foreign antigens, which will then be able to be picked up by what are called APCs or antigen presenting cells that are either circulating or within the tissue. So let's say a kidney transplant, there will be some dendritic cells, endothelial cells, renal tubular cells that can act as antigen presenting cells. And there are some that will infiltrate the graft from the blood. And these antigen presenting cells, their job is to ingest the peptides from the foreign tissue or whatever tissue they're looking at, break down those peptides into small molecules that they then express through their MHC or major histocompatibility complex seen over here to the T cell, which typically is a CD4 positive T cell. This signal pathway that you see on the left is called signal 1. Alone, it is not sufficient to give direction to the CD4 T cell as to what the next steps are. For that, the quality and the type of signal 2 becomes important. And there are a variety of signal 2 pathways. I've just shown over here one of them, the CD28B71 pathway, because that is where drugs like belatacept work. And the other drugs that we typically think of don't interact with this pathway just as yet. So let's say a CD4 T cell has been primed towards activation. That cell, depending on what are the cytokines in the milieu around that area, will then further differentiate into a variety of subtypes of TH cells. So there's TH1 or T helper 1, TH2, TH3, and then there are other types, TH17, and then what are called T regulatory cells. Each of them have specific cytokines that induce the differentiation to that cell type. And then each of those cells have specific signature cytokines that they will release into the cytoplasm. Now, the challenge is when you're looking at novel biomarkers that viral infections or any infection is really what the immune system was meant to detect. Infections are much more common for an immunocompetent immune system, and the placement of foreign allograft is not something that nature had anticipated. So when you're using the same pathways to be able to detect infection, some of those infections, moieties have actually developed mechanisms to evade immune detection. So for instance, effector CD8 positive T cells might use the fast ligand pathway to be able to attack the cell of interest that they consider to be infected. And these various viral infected cells, what I'm showing here is Epstein-Barr virus, just as an example, has learned to be able to block that fast ligand pathway over here by a variety of techniques, including by expressing something called PDL1 or programmed cell death L1, which binds to PD1 on the effector cells and induces them to go into an exhaustion phenotype. The same virally infected cell can secrete either viral IL-10 or utilize the human gene mechanisms to secrete a human IL-10, which then would promote more T cell regulatory activity. This is relevant because these biomarkers then might pick up infection as in a problem as well as rejection, and you will see some of the sensitivity and specificity around that. So the first major paper that I wanted to talk about has actually, it's been a while when it came out, and this was what was called the CTOT-04 study. CTOT stood for Clinical Trials in Organ Transplantation. And these studies were set up back in the 2004-2005 time point. And it took a good 10 years for them to actually do the 7-8 years worth of study, and then the data analysis, and then the paper writing and publication. So this group published this paper in New England Journal of Medicine, and it looked at the urinary cell mRNA profile and how that associated to acute rejection. So they had a variety of different molecules that they tested: CD3 epsilon, perforin, granzyme B, and IP10. Each of them had a higher difference. All of these scales that you see on the y-axis are logarithmic scales. So on a log scale, even a difference from 0 to 1 can be considered quite significant. And so what you see in these box and whisker plots are the changes that occurred in acute cellular rejection, which is in all of these graphs, it's the left bar, and then versus no rejection, or with stable function, but may not have had a completely clean biopsy. So each of them, the acute cellular rejection had a change that was probably at least 1 log-fold difference compared to the other states. And then when you plot an area under the curve, which looks at the sensitivity versus the 1- sensitivity, or as represented here, true positives versus the false positive fraction, in their original derivation cohort, they had an area under the curve of 0.85. And when then they went to their validation set, it had an area under the curve that dropped a little bit to 0.74. These would be considered good, not necessarily great. Ideally, something in the 0.90 range or above would be considered great. With that urine test, a variety of other blood rejection marker tests were also being studied. And we participated in one of the first pediatric CTOT studies. Ours was called CTOT-C, And this CTOT-C study that was done between 2004 to 2010 led to many different papers. It was a steroid avoidance versus steroid maintenance randomized control trial, but we also tested a peripheral blood diagnostic test for acute rejection, and that was published in American Journal of Transplantation 2012. It was a 5-gene expression panel. And then a variety of other groups. So Minnie Sarwal had led that group of our investigators for the 2012 paper, and then she has continued working on that with something she called a common rejection module. And then other groups have looked at other multivariable gene expression targeting panels. And these kind of things have been done for a variety of different situations, not just acute rejection, but even for fibrosis or for the chance that the rejection will respond to treatment. So the major paper that came out in blood gene expression profile was actually not in kidney transplantation. It was in the heart transplant world. And in that group, this company called XDX, which is now called CareDx, had created a panel for heart transplant, which was first studied in something called the Cargo study, which again showed, like what I showed you earlier for the CTOT-04, that there was an elevation in their markers with a higher score in the patients who had heart transplant dysfunction versus those that didn't. Now, this study that I'm showing you on this slide is called the IMAGE study. And this was the only randomized controlled trial that I know of where the gene expression panel itself was under test. So the two groups that you see on the graph, go back. The groups you see in the black and red, the group in black had routine endomyocardial biopsies. Since the heart transplant world does not have a blood marker like creatinine the way they do, that we do, they have to actually do these endomyocardial biopsies very frequently, which is, of course, highly invasive. The group in red did not undergo those endomyocardial biopsies. They underwent this AlloMap testing, and then clinical decisions were made based on whether the AlloMap score was elevated or not, and that might include an endomyocardial biopsy after that test. And what you see here is that in terms of the primary event, the two groups pretty much overlapped, the red and black lines in the bigger graph, and in the inset graph as well. When you look at the 2-year event rate, they're almost the same across the board for the routine biopsy or the gene expression profiling group. So there's other panels that are available now, and just trying to make sure that you see it in a balanced way. TrueGraph is from Transplant Genomics. And here what they did, this company used a non-biased approach. So they did actually whole genome sequencing and looked at thousands of genes to try to judge which of them were actually upregulated, which were downregulated, and which were not affected significantly in situations where the graft was in a quiescent state versus those that were in some form of dysfunction, where they did not necessarily classify what that dysfunction was due to, but just that the graft was not in a healthy state. So TX is the term they use for transplant excellence, not TXs. That means the graft has some problem going on. And what you see in the schematic diagrams below is that the test requisition form, then they send a kit to the center draws the blood in using that kit, ships the sample in the box they provide, and then within a 1 to 2 day turnaround, you get a report which will either say TX or not and may give you a value. So what happened in this development of TrueGraph is the molecular signatures were discovered through this unbiased approach. They looked at the 2,000 plus genes, and these were across 60 different pathways. And eventually, using the heat map representations of different types of kidney biopsies. So they're looking at 1,000 genes across 350 kidney biopsies. They then matched it to the histological analysis to develop these phenotypic morphological parameters. Now, you don't know actually what is in the TrueGraph panel, which genes, that has not been revealed. But the studies of TrueGraph were done through another CTOT study called CTOT-8. And here what you see is that when CTOT-8 was published, it had an area under the curve of 0.85, very similar to what the original derivation cohort had with CTOT-4. Now, these are molecular panels. You need to do some technological testing with that, and the technology is what is novel there. But Another group has said, what if we just took our clinical data that we have already in front of us and looking at the very large amounts of clinical data that we have at our large transplant centers, could we come up with some parameters using artificial intelligence to be able to use those parameters at, say, year one and tell us what that graft is going to do in terms of the next few years and how long it is going to survive? So this is work that Dr. Alexander Lupi and others have performed at the Paris Transplant Center, and they continue to work on this quite a bit. This particular graph I'm showing you is from their paper that was published in the British Medical Journal in 2019. And what you see on the left, the IBOX stands for integrative box, and then it provides a prognostic score that looks at these variety of different parameters. So whether there is an anti-HLA DSA, what's that mean fluorescence intensity? Whether there's proteinuria, what's the GFR at the time that this evaluation is being performed? And then on the biopsy, what's the glomerulitis score? What's the peritubular capillaritis, interstitial inflammation, tubulitis, and transplant glomerulopathy score or fibrosis scores? Putting all these together, they're able to create a prediction panel which tells you that this graft is going to not survive as well, or another graft is going to do very well and could survive extremely well, provided the patient continues along the same track. So I'm going to move now to another type of novel biomarker, which I will spend some time on, cell-free DNA. Here, the concept is fairly simple. All tissues in our body actually have a continuous cycle where some cells are dying and new cells might be formed. The cells that are dying will release DNA in a fragmented form into the bloodstream. And so we all have some circulating cell-free DNA in our bloodstream. And in a patient who has a foreign allograft kidney transplant, a small fraction of that cell-free DNA will be coming from that allograft, so will not match our own intrinsic DNA. And so what you can do is actually look at the different types of DNA being expressed, and just looking at the proportions, most of this is going to be from the host or the recipient. So if you exclude all that, then you're able to look at what might be foreign without even knowing what the donor genotyping is. And then you can see what that percentage is relative to the one that you excluded. And now you have a measure of allograft injury. So again, the concept here is you draw recipient blood into a special tube. In this case, it's called a Streck tube, which again, the company sends in a kit box. Then you send the tube back and the company then performs the analysis. They look at differences in the single nucleotide polymorphisms between the ones that they deem to be the recipient's host DNA versus that they deem to be foreign. And then they're able to calculate an absolute copy number, and they're able to calculate a percentage. What they also need to know is the degree of relatedness of the donor to the recipient. So for instance, if it's a sibling or if it's a parent, then they need to know that in advance. Now, this graph that you see over here is from the DART study. This was a prospective multi-center observational trial that was sponsored by the company across different kidney transplant centers. And they collected about 107 biopsies. 80 of them, as you can see on the left graph here, did not have any acute rejection on the biopsy at the time of cell-free DNA check. And then in the 27 acute rejection biopsies, you can see that the median cell-free DNA level was much higher than the median shown on the other graph. And the range, as well as the interquartile range and the upper limit, were also higher. In contrast, the serum creatinine medians actually were not that different between the two groups, and there was no statistical difference. This p-value is 0.23, whereas it was highly significant here. So again, areas under the curve, true positives versus the false positive fraction. And here you can see them represented. The number that came out was about 0.73. Here you can see on this bottom left here is that sensitivity and specificity cutoffs in general, and true over here too, as you increase one, the other will drop. And so the cutoff, where is that point where it crosses, was right about here. And that's where the area under the curve came. So at that point, the sensitivity is going to be about 59%, but the specificity is much higher at about 84%. And what that leads to is a situation where you can have higher NPVs at the lower values. So when you're running a value that instead of being 1% is say 0.2%, you'll have an extremely high NPV as shown in this bottom right graph. In this graph from the same paper, what was noticed was that antibody-mediated rejections, or ABMRs, had many more of these bright red or dark red squares. T-cell mediated rejection, which you see in the middle over here, had more of the lighter reds and not as many of the dark. What I want to also bring to your notice is what happened with BK virus, which is shown right over here. There were 5 samples, and 3 of those 5 actually had elevations in the cell-free DNA, including one that looked to be on the much higher range. So this is the cutoff here is 1%. And as you're going further and further away from that 1%, you're reaching values that could be in the 3–4% range. So the discrimination of cell-free DNA has also been studied by other groups. And this is from Tara Sigdal and again, Mini Sarwal. And they showed very similar pattern of donor-derived cell-free DNA in acute rejection versus other categories such as stable or other types of injury. And the EGFR scoring also showed that difference the same way. What this paper also showed is how the cell-free DNA levels could change prior to a biopsy. So what you see in the gray line, dashed line, are the cell-free DNA levels. Rejection status is represented in red in circles, acute rejection, whereas non-rejection are all the blue circles. And then biopsy false or true is represented in either the black circles or the black triangles. So a lot of these graphs, we won't go through all of them. You see a lot of blue circles, and you see that level of the cell-free DNA going up or coming up relative to its prior value in that same patient. And the red triangle comes a little bit later in the course. Often the elevation in cell-free DNA precedes that. And this is an important point when we want to talk about what might happen in surveillance. So I've shown you 2 different types of cell-free DNA tests. There are actually now 4 on the market. One paper has looked at whether these were comparable. And what you see is if you're looking under the 1% level, which is the top left graph, while there is a line of identity along this which you can create, but many of the circles do spread quite a little bit distance away from that line. I would have liked to see them line up a little better. And then when you get above 1%, you can see again that the circles deviate from the line a fair bit. So these suggest that these tests are not as comparable to each other as you might think. And so generally, if you're going to monitor, then stick to the same test. And in the US, we have AlloSure, we have Natera's Prospera test, and then Virocor has a test called Virocor Track. And then there's a new one I just heard of a few weeks ago called Vitagraft. So far, everything I showed you was related to percent cell-free DNA. There is a way to measure the absolute levels. That's actually what gets started. Companies don't typically report this, but some of the groups have been able to go back and obtain the absolute levels. And then they look to see whether there were absolute levels versus percentage discrepancies. So what you see in the graph on the right is the copies per ml on the y-axis and the percent fraction on the x-axis. And these 2 blue circles that are highlighted in the red oval, that shows you where the results were discrepant, where the copies would suggest a patient actually had an elevation in the copies, but the percentage of cell-free DNA was less than 1%. So this group actually proposed, both these groups, to have a 2-threshold algorithm and use both these things to see if either goes up, then to be concerned. If both go up, to certainly be concerned. Now, everything I've shown you so far was in the adults. I'm a pediatric nephrologist, though I work in this field quite a bit. And we have done some studies of pediatric cell-free DNA. And I'll show you some of the studies that have come out so far. The reason we were interested was that in young children, we don't actually size match and place a pediatric kidney into a pediatric recipient. When we did do that back in the 80s, 90s, we realized that the small blood vessels to the small blood vessels increased the thrombosis risk by about 8-fold. So what we do is we choose an adult kidney, not for the nephron mass, but to get that bigger blood vessel. And then in the very young children, those adult kidneys are anastomosed to the biggest blood vessels of the infant or toddler, which would be the aorta and the IVC. And that is how we have been able to reduce the thrombosis risk quite a bit. In the greater nephron mass that you get with that, though, the challenge was, would this 1% cutoff still work? Because now there might be so much more cell-free DNA coming out from that bigger kidney that's sitting in that small child. So 3 studies have been done. Dechu Pulianda, who's at Cedars-Sinai in Los Angeles, published this study in pediatric transplantation. That particular center gets a lot of patients who are highly sensitized, and they are at high risk for antibody-mediated rejection, both in the adult and pediatric world. So they studied 67 pediatric kidney transplant recipients, but many of these, because they were sensitized, their group had a lot of antibody-mediated rejection. There was only 3 T-cell mediated rejections in their group of 29. And what they showed was that the cell-free DNA level of 1% actually worked fine. 86% sensitivity, 100% specificity, and area under the curve was amazing at 0.996. Another group in England and Great Ormond Street Hospital, they have created their own in-house assay. Europe, the assays that are commercially available have not yet penetrated quite as well. So there they looked at a bunch of 159 pediatric solid organ transplant recipients, of whom only 44 were kidney, the rest were other organs. And what they looked at was the donor fraction percent, and their cutoff was 1.29%. And then the concentration, which they showed as 0.47 copies in the kidney world. They had different donor fractions in the other organs, and that's a whole separate topic in case anyone's interested. They had a high specificity and sensitivity, 97% each at these thresholds. We had the opportunity to use some of our biobank samples. So for this particular part of the analysis for the T-cell mediated rejections or antibody mediated rejections, we used 109 biobank samples from 54 pediatric kidney transplant recipients over an 8-year period. This was published in CJSON 2022. And what we found was that the donor-derived cell-free DNA was elevated in the biopsies where there was rejection versus where there wasn't. Versus serum creatinine that really on the top right, you can see over here, hardly had any change. And area under the curve for us was 0.82 versus for serum creatinine, something like 0.50. And so the donor-derived cell-free DNA was highly significant. So all of these studies suggest that 1% or close to it would actually be fine. And why is that the case? Our hypothesis was the opposite. And it may be that the turnover is different in the baseline level. And so both the numerator and denominator may be changing. So here's our cutoffs at this different 1% cutoff using the adult. We had a specificity of 96% or 90%. Sensitivity is low in our population. The challenge with these kind of tests with cell-free DNA or with these blood gene expression panels is this issue of BK virus nephropathy. And you see that over here that you can actually see an elevation in those, not just with acute rejection types, but with BK virus nephropathy types. And depending on what you're looking at, your training data, it had an area under the curve of 0.93. And in the validation cohort, it drops. This is true in many studies that the validation cohort, whatever test you're doing, as you have a wider population that's more heterogeneous, the accuracy of the test can drop. So we, again, using our biobank samples, looked at what happened with BK. And we have been actually in our center doing BK in the urine first before we switched to blood, only after the BK in the urine turns positive. So we were able to see that in patients who did not develop viruria or viremia, which is the dark black box, the cell-free DNA levels stayed low. They went up to their peak at the onset of the viruria or onset of viremia. That's the gray boxes. And then as you treated it with various interventions, and as the BK viral levels dropped, the cell-free DNA levels dropped as well. So it's interesting that even at a viruria stage, which you wouldn't expect necessarily graft injury, these cell-free DNA levels suggest that some graft injury is occurring. And what you see over here now is a different test called QSANT. And this is from groups that have looked at urinary biomarkers that include the cell-free DNA, as you see over here, but have also added methylated cell-free DNA, clusterin, a chemokine called CXCL10, the urine creatinine, and the total protein that's measurable in that urine. And using these 6 biomarkers, they were able to create a Q score, which is from 0 to 100, and that reflects the probability of allograft rejection risk. These studies were done on about 600 patients, and then they were primarily looking at acute rejection versus stable. That's what you see in the sensitivity and specificity graphs that are on the bottom. And what you can see is how the Q score performed versus other things like EGFR or protein creatinine. And on this one, in the B graph, what you see is adults versus pediatrics. Adults do a little bit better, almost close to 100%, whereas pediatrics, it's a little bit lower than that. So impressive results in general, and these need to be validated by other groups, and this is something that I think the groups are working on. Now, there's another concept of exosomes. An exosome basically is an outfolding of the cell membrane, and it basically inverts whatever antigens are expressed on the cell membrane. Now they get expressed on the inner lining of these exosomes, and they're released into the circulation. You can detect these in blood. You can also detect them in urine. And so groups have looked at these kind of vesicle numbers and what they're carrying inside the vesicles to see if this could be a biomarker for acute rejection. Again, I think this is probably more for any type of graft injury. So the area under the curve here, you can see is 0.865 on this side, 0.93 on this side. So I've told you that these different biomarkers, they elevate with rejection, but they could also elevate with BK virus. And we have seen that they elevate with urinary tract infection too. So how do you differentiate those? Long back, before these novel biomarkers made it here, people had actually thought about this with some of the older tests, like the immuno test, which was more an infection marker, CD30, which is more a rejection marker, and the PRA class 2, which would reflect antibody-mediated rejection. So this group, Israeli et al., compared these different markers to try to create zones where they said, if you look at them in a combination rather than isolation, you can judge which is the stable patient versus which has an acute cellular rejection. And that actually was a paper that influenced us quite a bit. We had the opportunity to compare 2 markers, one reflecting rejection, which was the indoleamine 2,3-dioxygenase. And we compared that to, in rejection and infection, to another test, which was the CD4-ATP, which you see over here. And the IDO is actually not measured directly, it's an enzyme. What you measure is the substrate and the product. Kynurenines are the substrates and tryptophan is the product. Sorry, reverse way, tryptophan is the substrate and it gets degraded into kynurins. So the IDO levels worked as a decent biomarker for acute rejection. The CD4-ATP levels worked as a decent biomarker for infection prediction. And we were doing this longitudinally in different patients. We actually do 2 different types of sets that accounted for or did not account for within patient variability. And then the combination of the 2 were even better than looking at any one test alone. And that's what we had hoped would become the norm in the practice. So similarly, the group that has performed one of the cell-free DNA tests from the CTOT-8 study, once the gene expression test panel came up, in this case, they were looking at the TrueGraph panel, they wanted to see if they could go back and judge if the combination of the 2 was better than either one alone. So what you see in this visual abstract is the gene expression profile assay, their negative predictive value and the area under the curve. And then what happens to the cell-free DNA level in isolation. And when you combine the 2 tests, and you see a decent area under the curve with each test separately, and it goes up from 0.7s into the 0.81 range when you combine these together. Now, I want to spend a couple of minutes on the logistics of doing these kind of novel tests. You have to get special blood sample kits, which are supplied by the company. And then there is the blood volume involved. If you're having to send 2 tubes for the PAX gene for the gene expression and another 2 tubes of 5 ml each in the STREC tubes for the cell-free DNA, that's 20 ml of blood that you have to send to these companies over what you have already sent for your serum creatinine, CBC, renal function panel, etc. So that can add up to quite a bit of blood volume, even for an adult. And for a child, that could be prohibitively big, especially if you have to do this repeatedly. Now, the turnaround time these companies have managed is usually quite short, 1 to 2 days, so that does not become a restriction. I wanted to emphasize to you that these tests, all of them developed an analytical validity. That means they were able to show, the companies were able to show to the FDA that if you take blood tests, the result you get once is the same result you'll get if you wait a day or if you split a sample. The assay works well. Clinical validity was not something that the FDA had focused on until recently for tests rather than for drugs. And so clinical validity means, does this test actually tell you something about the nature of what might be happening to the patient? And in the old immunotest, what happened was the company went to the FDA and got the analytical validity FDA approval. And then they went and said that the test is FDA approved. That didn't necessarily tell us about clinical validity. We had to figure that out ourselves. And it didn't tell us about the context of use. So any test, if you draw it across a general population where 99% of the population may not ever have a transplant, then how do you know that result is going to be meaningful? The best meaning comes when you send it in transplant recipients, and you send it not only in that group, but when it's particularly indicated. And that's how your performance of the test increases. So that is what is context of use. Surveillance biopsies would not necessarily be as good context as for cause. Now, what the newer companies have done is, rather than going to the FDA for approval, they went to the central insurance company in the United States called Medicare and were able to have Medicare set a reimbursement rate for tests. And some of these blood tests are getting reimbursed at $2,800 per single test. The way they can get away with that kind of money is that the biopsies themselves that you might be able to avoid cost in the range of $10 to $13 per USA centers. But when you want to do this test multiple times in surveillance, $2,800 per test adds up quite a bit. So my conclusions and current thinking so far is creatinine rises late. We do need these earlier biomarkers. And many of the current tests, the ones that are novel and coming out, they can help predict graft stability, and they may be able to predict injury as well. But the nature of the injury is a little harder. It could be TCMR, could be ABMR, antibody, urinary tract infections or viral infections. And particularly the opportunistic viruses such as BK virus, their loads are challenging because when you look at that viral load, my assay is going to be different from your assay and might be different from a central lab assay. Each of these assays are not standardized. Their cutoffs are different, primers different, strength of the immune response is different. So it's not possible to say over a period of time that I did one test and my patient did another test locally and I'll be able to compare. The panel approach that I've discussed may be another good way of trying to do this way of addressing. Right now, what some of these companies are doing is they're measuring the BK in the urine when they're doing the Euromap test. And if they have a high Euromap level or score, then the BK value is reported out, and then the physicians are told that this is also potentially a cause. Annulloviruses are another group of viruses that are non-pathogenic as fast as we know right now. And a group at Stanford showed that those viruses bloom as you do transplant immunosuppression. So we have looked at this in our own hands. We have not seen a good association with annulloviruses, but we haven't done this as a prospective trial. Other groups in Austria have, particularly Grigao bonds. They seem to have good results, which we have been struggling to replicate. So with that, I will stop over here and leave some time for questions. I wanted to acknowledge a variety of colleagues and authors, such as Dr. Malone, who I'd mentioned in other readings. Here's some of my contact information, email or Twitter or website. And I wanted to introduce this last thing to you. Just this is our group on the top right here of the various doctors who are senior doctors or in training, plus the nurse practitioners and our nurses that are part of the group. And then we have recently obtained a Pediatric Center of Excellence in Nephrology through a special NIH grant, which is under open competition every 5 years. And so this year we were able to be in the top 3 and obtained this particular prestigious grant. The science in this grant is completely different. Here we're trying to develop a molecular and genomic atlas of the pediatric kidney in both health and disease. But I just wanted you to be aware of this Center of Excellence that we now have. So I will stop over here. Thank you for your attention, and I'll be happy to take any questions.
Thank you very much, Dr. Vikas. That was really a great talk. I think it simplified and introduced us to the concept really that we could use biomarkers, not just for the diagnosis of acute rejection, but also for surveillance. So I had a question to ask you, a practical question, and then we actually have our junior faculty with us who we were going to tell you about a patient of ours and see how you thought these biomarkers might apply in our setting and whether they would. So my question to you was actually, how do you use these biomarkers in your clinical practice? Because we are not yet, we don't have access to them and we haven't started using them.
Yeah, so since we have been doing a variety of research studies, the biobanking studies were retrospective in nature, but we have been funded through the company and through society foundations to do prospective studies. So one of them, which we call POCRA, is actually in its final year now. So we'll have the data by the end of this year, and hopefully we'll be able to present and publish that by early next year. And that's where we're doing these biomarkers in association to the surveillance biopsies. And another study that we have that is in our own center, we're looking at what happens to these biomarkers after the first year. Most of them get tested in the first year post-transplant. So those results we'll have a little bit later. Coming to your question, so we have not yet gone to the clinical practice realm in pediatrics because there are all these research studies that we want to complete, and we want to fully understand the context of how best these markers can be used. So for the American Transplant Congress that will come this year in June, the deadline abstracts was in December. We submitted some abstracts related not to the cell-free DNA, which was what are the studies I just mentioned, but the AlloMap kidney, which is gene expression panel, and now the Euromap, which is the urinary gene expression panel. We've got some abstracts there, which suggest they will do well, but we want to study them a little bit more. On the adult side, many centers have made this a clinical practice. I don't know that many of them are doing this 7 times in the first year testing. There is a fairly large amount of blood volume that is needed, and it has to be at certain specific schedules. So those are the kind of challenges in having to do it. But I know of many centers in the US that have moved to that. I don't know of any centers yet that have gone away from creatinine and replaced this with creatinine.
Okay, great. Soumya, are you here? So Dr. Soumya
Yes,
Reddy is our recently graduated DM resident, and she's doing her year of senior residency with us.
Hello, Soumya. Go ahead.
Hello, sir. Thank you so much for such a wonderful talk. It was really great to learn about the whole wide spectrum of biomarkers that are available. My question was more a clinically related question. So we have this 15-year-old girl who's completed 9 months post-transplant now. Her native kidney disease was of juvenile nephronophthisis, but not genetically confirmed. The donor was her mother, who had no comorbidities. She's a 32-year-old lady. Pre-transplant HLA typing showed around 6 mismatches, 1 each in HLA, B, C, D, RDP, and DQ. And the lysate crossmatch that was done twice prior to transplant showed no evidence of donor-specific antibodies. As per our unit protocol, we went ahead with induction with basiliximab and continued her on triple immunosuppression with steroids, tacrolimus, and mycophenolate. In the immediate post-transplant period, in the first 3 months or so, she had recurrent episodes of urinary tract infections. She had around 3 such episodes, and during each episode, her creatinine would rise up to a peak of around 4, and then would come down to around 1 to 1.5 after completion of the antibiotic course. So the lowest creatinine that we had beyond the 3rd month of transplant was around 1.14. And this remained stable for the next 3 months or so. And she was on regular follow-up with us with adequate tacrolimus levels and good compliance to medications. So the question is that she actually came to us yesterday and was admitted with rise in creatinine to 7 that was noted yesterday, and she was absolutely asymptomatic. No evidence of any infection. Previous episodes of UTI that she had, we ruled out. She had no feature suggestive, clinically at least, of graft rejection. She was supposedly compliant to all her medications. She had adequate tacrolimus levels. So we were very surprised with the report. Didn't really know why we were faced with such a problem. So my question is, could we have prevented this? Could we have used these biomarkers that you've spoken about to probably pick up what was going on much earlier? And a follow-up question is, if we do use cell-free DNA, how often do we actually monitor these levels?
I wanted to add, Dr. Vikasa, we're just getting ready to biopsy her tomorrow. We have to stabilize her first. We don't have a biopsy ready. We do, as hot off the press, just found that she has class 2 DSA positive, so most likely she has rejection.
Yes. So thank you for that, Soumya, and thank you for the questions. I think on the chats, people are asking similar type of question about what is that, when to do these and recommending. So let me start first with your patient. A jump to a 7, this suddenly creatinine, you think acute rejection is right top of your list, most likely antibody mediated. The fact that you now see a class 2 DSA could have come even in a compliant patient, though you would think that is less likely. I will tell you that my patients who tell me that they're compliant, sometimes I wonder about them. You mentioned your patient, I think is 15. And my teenagers, I have stopped asking them, are you taking your medications? I ask them directly, how many have you missed in the last month? And maybe a couple will get offended with me at that question. Most of the others look at me sheepishly and will give a number. And then I have to multiply that by number by some unknown factor to try to judge how many they truly have missed. So with teenagers, I have unfortunately become this cynical, skeptical person in terms of the answers. And we do see compliance drop in the later part of the first year or just after the first year. We did, in fact, medication adherence studies, the Take It 1 study, which we published. This is a whole different series, and Take It 2, which is now just completed. And in Take It 1, we were monitoring their adherence through electronic pillboxes on an everyday basis through a whole period of 15 months. And the group that got automated text messages, those maintained a decent level of compliance across the group. The other group, which just had the pillboxes, there was a gradual fall in compliance over that year. So I think that's what's happening. Now, coming to your question of how to use these novel biomarkers, if you do it in a for-cause basis, here you already have your creatinine that's elevated at 7. You're going to biopsy this patient irrespective, even if you had unless you had severe symptoms suggesting a pyelonephritis and like 200 WBCs in a urine, then you might wait for the biopsy till you've treated the UTI. But in any other circumstance, you're going to do the rejection biopsy. Even the BK test will take time to come back. So it's really when you have a subtle elevation in creatinine, let's say you went up from 1 to 1.2. Now, is this within the range of lab error? Is this a real change? And your creatinine may not be sufficient to tell you that. The tacrolimus level might be okay at that point in time, or CNI level. And then you're likely to biopsy if you're worried still. Here, you could use a cell-free DNA level in isolation or a gene expression score. And if that score is normal, or if the cell-free DNA is low, then you might say, I don't need to biopsy. On the other hand, if your score is high, it's not going to tell you the nature of the lesion in the patient. You will have to biopsy then. So it's not necessarily avoiding a biopsy in that situation. Now I'm going to switch to what happens if you're doing surveillance testing. And of course, these companies would like you to do surveillance testing because now you're doing it more often and it makes more money for them. Whether surveillance is beneficial, I think we need real data for it. And there are a couple of studies in the cell-free DNA world, the Okra study and the Admiral study that have been recently published, which do suggest some benefit of surveillance. One of the slides I showed you showed how these tests can elevate prior to a biopsy showing an actual rejection or prior to a creatinine. What is the optimal interval between surveillance tests is really hard. Again, just based on one of the companies, and this is CareDx, which has given a recommendation out there. I don't want to say that I'm endorsing any recommendation because we don't have enough data, I think, to do that. But they would like you to do this monthly for the first 4 months. And then I think they said another 3 tests between month 4 to 12. So probably every 2 months then. So 7 tests in the first 12 months and then quarterly for the year 2 and year 3. Is that the right schedule? I don't know. And will that apply to the gene expression panels, either in blood or urine? Again, we don't know that yet. So that's the difference between how you might do it on surveillance versus how you might do it in a for-cause basis. Cost is going to be a big factor for that. If the test is as expensive as right now these are, surveillance could be a problem for you. You might save one biopsy, but then you're going to add up so much more cost there. So sorry for the long answer, but I wanted to cover all the different aspects that were coming in your case, as well as what is being was being asked in the chat. I think there are a couple more questions in the chat.
Yeah, you'd like
Yeah.
me to read them? So Dr. Swarnalata was asking biomarker which can differentiate the type and stage of rejection, a T cell ABMR, active chronic, so that could help with therapeutic decision.
Yeah, so it's interesting that the cell-free DNA levels seem to go much higher when it's an antibody-mediated rejection and not quite as high in T-cell mediated rejection. And in the gene expression panels, what we have seen in some of our early data, which is what we are going to hopefully present at the American Transplant Congress, we've seen that the scores seem to elevate more in cell-mediated rejection. And that was, in fact, what was the reason why they wanted to combine those 2 tests in one of the slides that I showed you. So these 2 tests might have some overlap, but if you do them together, it's possible that you can get a better sensitivity and specificity. And then looking at the relative elevations in one or the other might give you a clue as to which type of rejection you're looking at. I don't know that they would replace a biopsy differentiation of the type of rejection yet. I think we're not quite there at that point.
Go ahead, Priya, you have a question?
So my question actually was, there's one in the chat box, but just is mentioned as a donor-derived cell-free DNA. So does that mean that, and you had mentioned that the degree of relatedness does need to be shared with the testing lab. So is this a test where the donor HLA details and I guess the gene details need to be known?
No, so that is the advantage of these tests that have come out, that they have managed to get away from the need for knowing the donor genotyping or getting a donor sample. If you have a deceased donor, how would you get that easily? And so what they're doing is they're making an assumption that the overwhelming majority of your DNA that's going to be present in that is going to come from the recipient or the host. And then whatever is left, that fraction, which could be 0.1 or 1 or 2%, that's foreign DNA is the only one they have to concentrate on. And the degree of relatedness does matter because as you're trying to not genotype, but as you're looking at these polymorphisms, they need to account for that. Siblings, parent, for instance, a parent is going to have half the genotype is going to be identical. And they have been able to make some changes. When we get our reports, we get a raw value, and then we see an adjusted value based on the degree of relatedness. Each company uses some proprietary algorithm for that. I think in the question and answer, there's another one here. Somebody's asked, Dr. Sahai has asked whether these biomarkers will be clinically useful. The biopsy still is the gold standard, and biomarkers have been around for a long time. So really an excellent question. 2 points here. The biopsy, unfortunately, is not a gold standard because even the biopsies have significant limitations. So in fact, our pathologists say that it is a bronze standard rather than gold or silver because of these limitations. So when we are testing these biomarkers, we are testing them against an imperfect standard. So when we see an area under the curve, which is 0.7, 0.8, how do we know that it might not have been higher? Because the biopsy missed a rejection too. And that is the challenge with this. The biopsy might actually be superior in some situations, or the biomarkers might be superior in some situation. So yes, biomarkers have been around a long time. The CTOT04 study started in 2004 from studies that had already been published in the New England Journal in, I think, 2001. And those were urinary profiles of gene expression, and only now they're being commercialized That has taken about 20 years for that to happen. So I think there is now suddenly an acceleration of progress. I think you'll see a lot more of this in the next few years. Part of this now will relate to how the logistics and cost can be managed by these sponsors.
I think
Dr.
Dr. Manisha had another question asking if you routinely do tacrolimus gene polymorphisms in clinical practice.
Great question again. Thank you. We do not. These gene polymorphisms for the tacrolimus, I presume this is for metabolism to find out if they're slow or fast metabolizers. I don't know that many centers do this. The data are there that you can judge a fast or slow metabolizer and you can adjust the target levels accordingly. But again, this is a complicated test to do and therefore has not been routine practice. Certain ethnicities do show variations in this, and ideally we'd like to get to a personalized medicine where we do this on a regular basis, but not in our centers either on the adult or pediatric side. Then Dr. Vala has a question about whether this cell-free DNA has any role after 3 years. I don't know of data about that. Again, this is an excellent question about, suppose you get a late antibody-mediated rejection, would the cell-free DNA pick it up then? Theoretically, it should. The companies haven't done these studies. I don't know of individual investigators that have done them as yet. It could be something that people are studying. We are looking at what's happening in the 1 to 3 year period, but not right now at what happens post 3 years. So again, something I think we should look forward to seeing what data come out for that.
So Dr. Vikas, I had a question that is quite close to what Dr. Manisha has just typed in the question and answer box. So if the scenario here is that we, majority of our transplants are live related. I'm talking about children, and I'm sure it's the same in adults, live related transplants. And of course, nowadays we do ensure that induction therapy is a mandate. There was an era that we would leave it to the parents and give them a choice because it's expensive, but now we prepare them for that. And we are relative on a lower immunosuppression compared to the West. Again, because of cost issues, and most of these families spend from their own pocket and there's no insurance and so on. So in that scenario, we don't have protocol biopsies. And like she just mentioned, if you have a creatinine which is normal or near normal, like the girl who came a month back had 1.1, we know it's not normal, but we are not sure whether we need to jump and do a biopsy. Is there proteinuria? In some scenarios, we may have 2+ proteins, and we quantify that, and it may be significant. So in this scenario, if you do a biopsy and it is normal, where does the biomarker have a place here? And in the second scenario, if you do a biopsy, and for example, I remember one of our kids in the past had a transplant glomerulopathy with an MPGN pattern, had proteinuria. So it was too late. The family, of course, for other reasons, did not want to go ahead with any aggressive treatment. But so proteinuria, near normal creatinine, biopsy being normal or showing some changes for the future. I know that we are not using these, and probably in my time we'll never use this in our center. But where do you see the role of these biomarkers?
So in these situations where the creatinine and biopsy are telling you one thing and the biomarker might be telling you something different, what we have found through some of our studies, there are other situations where the biomarker levels elevate, not just the urinary tract infections or the BK virus, which I've already mentioned. Surprisingly, recurrent disease or retransplants can have elevations too. So with the gene expression panel, what we found is the patients who had a second kidney and the first allograft is still in place, was not removed. So they're running a little bit higher score. So these are the nuances that are not obvious, and until you actually don't do them or people don't write about them, may not realize that. Now, if you don't have any of those situations, let's say your biomarker went up and your creatinine at this point looks okay, maybe you've not done a biopsy here. What I would do personally is I would follow that creatinine much more closely instead of having a long interval. Let's say the patient is 2 years out and you happen to have gotten this biomarker. Your next creatinine might be a month later in your normal situation, but here I would get another creatinine quickly, maybe in 2 weeks, because the possibility is also that this biomarker elevation is that first early detection of an immune attack occurring for a variety of reasons. And so let's say your second creatinine now has elevated further. Now you know that there's something wrong and that triggers a biopsy. But if your creatinine stays normal or comes back down in the back to its original baseline 2 weeks later, then I would just watch. So I am still using creatinine as an important marker, and I'm trying to see how that temporal change in creatinine relates to the temporal change in a biomarker.
And what about proteinuria?
How it changes with the biomarker?
Meaning the temporal association, is it that you would find a biomarker rise even before proteinuria becomes overtly significant?
In our own studies, we have not seen much of proteinuria, but most of our studies so far have been very early within the first year where there's extremely low levels. So I'm trying to remember what has been reported in the adult studies that have gone out to about 3 years. Admiral study has done that. So what they found, and I'm going to quote them the way I remember it, so I may be off. If you had more than 2 or more elevations in, say, the cell-free DNA in the first year, the chances of fibrosis in the biopsy beyond the first year were much higher. Now, whether they looked at proteinuria also in those patients, I don't remember that. I haven't seen much in terms of the association to subsequent proteinuria, but it's probably buried in some of those papers. So I'll have to go back and look, and I can get back to you on that question.
Are there some situations other than BK where you would get a false elevation or an elevation of the cell-free DNA not from acute rejection?
Yeah, so the bacterial urinary tract infections, pyelonephritis, and then the recurrent disease, and in some cases, not the cell-free DNA, but for the gene expression panels, the retransplants. So the other three, they elevate pretty much every biomarker, and it has to be a graft infection. So we also monitor for systemic CMV and systemic EBV infections. And what we have found is they do not elevate the intragraft novel biomarkers. They may elevate a lot of other things, but if they're not affecting the graft itself, we don't see an elevation in cell-free DNA there.
Okay. So it's just a more, I guess, a signal of injury to the graft.
And that is exactly the word I use, allograft injury. And even the companies, they are trying to promote it as a rejection test. But if you look at what's in the writings, they're saying allograft injury, and they have to be fairly precise about that. So in fact, nowadays, for instance, the urinary expression panels that the companies want to commercialize, they're not going to be reporting to you just the gene expression panel score. They're also going to do a BK virus test at the same time. And they're going to tell you this, here's the gene expression panel score, and here's the BK virus load, which could be 0 or could be elevated. Then it is up to you to interpret the two tests together. So they understand that BK virus elevates these test results.
So the patient has a high creatinine, there's no, you're still back to doing a biopsy.
Yeah, so that's the big question. Is this going to prevent biopsies? Is it actually going to generate more biopsies because you got an elevation in the test? And that's where we are also struggling in our community, trying to figure out what is the best context of use. So analytical validity, we know that this test, you send it once, you split it up, you'll get the same result from that same blood sample. Clinical validity, we have some idea, but context of use is still a challenge for us.
In general, what is the cost?
Well, so that in the US, $2,800 per test is an astronomical cost for a blood test. And that's what these gene expression panels, TrueGraph, or the cell-free DNA test, in any of those, that's what Medicare, the federal insurance, put as their reimbursement amount. So these companies didn't go to the FDA to get get FDA approval. They went to the insurance reimburser and said, tell us how much we will get reimbursed. Otherwise, the previous immunotest never really got traction because it was not getting reimbursed. So it's really how the companies approach the way of getting the test to the market. And now that there is a reimbursement, value that they get, they're not spending their own amount of money on the center or the patient level to get it. 2,800 though is the highest amount I've ever heard for a single blood test. A biopsy is 10 to 13,000 US dollars in different centers in the US, something in that range. So it's about 1/5 of the cost of that. But then if you do 5 tests, and if you still had to do a biopsy, have you actually saved money? Now, this test is, the cell-free DNA has been commercialized in Australia. They call it something different there. I don't know how they're getting it reimbursed in Australia. They have a single payer system where it's one national healthcare service. I don't know what they're doing in the UK. So this is going to be a challenge for all the countries with the different ways that either patients are still self-pay or there is some insurance coverage and how it's going to be covered.
I think one last question is coming from Dr. Manisha again. She wanted to know if there's any influence of the immunosuppression regime on biomarker profile. Say, for example, if you use a steroid-free protocol.
Actually, we have not looked at that, and we can, because most of our patients, but not all, go on to a steroid. What we call a rapid steroid taper is what we do is with just the first 3 days post-transplant, there's perioperative steroids, and then they don't see anything. And that is probably 75–80% of our patients. But the patients who have a primary glomerular disease or a complement-mediated disease or a retransplant or somebody who's sensitized, these patients, we have a steroid maintenance regime. So we actually have the ability. We have not gone back into our data and done that analysis. Thank you for that question. We'll go back and see if we see something different, and then we can report that out to the community.
I think we've covered all the questions in the chat, in the question box. So I think if there are no further questions, I will take this opportunity to say a very big thank you to you, Dr. Vikas. I know you're on vacation here in India, so thank you for spending some time with all of us and discussing this important topic. And hopefully we'll see more of literature and context about biomarkers in kidney transplant.
Thank you, Dr. Priya. Thank you, Dr. Aparna.
And thank you very much to Win India team also for giving us this opportunity to invite Dr. Vikas.
Thank you everyone. Thank you, Dr. Vikas, for joining us. It was a wonderful evening for all of us to learn on this novel aspect. Thank you, Dr. Priya, and thank you, Dr. Arpana, for moderating the session. Thank you so much. Good night, everyone.
Thank you, Win India.
Bye.
Bye.
Thank you. Bye.